Tamam hocam. Biz o zaman.
Hepinize sunumlar diliyorum.
Sağ olun hocam. Teşekkür ederiz. Sağ olun. Kolay gelsin. Evet değerli arkadaşlar. 4. Uluslararası Fen Bilimleri Uluslararası Öğrenciler Fen Bilimleri Kongresi'ne hoş geldiniz. Session 12'de bizim 6 başlığımız 6 başlıkta we have 6 titles and we will start with the characterization of plasmid from AntibioticRes Pseudomonas isolated from soil sample. And the presenter is Yunusa Umar Muazu is here. Uluslararası Öğrenciler Fen Bilimleri Kongresi'ne hoş geldiniz.
Hoş bulduk hocam.
Section 12'de bizim 6 başlığımız 6 başlıkta we have 6 titles and we will start with the characterization of plasmid from AntibioticRes Pseudomonas isolated from soil sample. And the presenter is Yunusa Umar Muazu. Is here?
Yes, I'm here.
Okay. Uluslararası Öğrenciler Fen Bilimleri Kongresi'ne hoş geldiniz.
Hoş bulduk.
12'de sesler tekrar bana geliyor ama
Burada da aynı.
senkronize şekilde gelmiyor mu? Benim konuştuğum anda gelmiyor mu sesler? Characterization of plasmid from AntibioticRes Pseudomonas isolated from soil sample. Acaba bu katılımcılardan özellikle sunumu yapmayacak olanların seslerini kapatmasını rica etsek. Sadece sunumu yapacak olan
Yes, I'm here. But I can't start the sharing, the screen sharing. It is disabled. I can share my screen. Hocam.
Okay. Can you try again?
Okay. Yes, it's okay now. Herkes merhaba. My name is Umar Muazu Yunusa. My name is Umar Muazu Yunusa. Today I'm going to present on characterization of plasmids from antibiotic resistant species isolated from soil sample. In this our research, we isolated plasmids from Pseudomonas species. Introduction. As you all know that antibiotic resistance, which can occur naturally or artificially, is a serious threat to global health and food security. A growing number of infections such as tuberculosis, salmonellosis, gonorrhea are becoming harder to treat as a result of this antibiotic resistance. The negative consequences of this antibiotic resistance include longer hospital stay, higher medical costs, increased mortality, and sometimes it compromises the success of major surgery. The mechanism of which antibiotic resistance affect our cell is as follows. As we can see that this, the blue cycle are the antibiotic. So because of this, when the organism develop an antibiotic resistance, it becomes harder for the antibiotic to insert into the cell. It can also increase the influx of those antibiotics when they enter into the cell. And sometimes it's or it can protect the target organism. In the second image, as you can see, the orange color are no-resistant bacteria, while the purple color are the antibiotic. The army green color is the resistant bacteria. So when antibiotic entered into the cell, it can kill the no-resistant bacteria, as you can see, the orange color, while the resistant bacteria, the green one, it can multiply. Pseudomonas species are aerobic non-fermenting gram-negative bacteria that has the adaptability to withstand several oxidative and nutritional stress. This organism has an important picture in production of substances such as enzyme, protein, polysaccharide, extracellular DNA, etc. The extracellular DNA, as you know, is often referred as plasmid. It's a small DNA molecule within the cytoplasm of a bacterium or protozoan cell that can replicate independently. The current study aimed to determine the resistance of this species, that's a Pseudomonas organism, against some antibiotic, and to isolate this plasmids from the bacterial species. In our research, we collected this sample from the Department of Biochemistry, Bayero University, Kano, Nigeria. So to isolate the organism, we prepared 3 different agar: cetrimide agar, nutrient agar, neutron broth. So we prepare cetrimide agar to isolate and to make presumptive identification of the Pseudomonas species. How we prepare cetrimide agar is as follows. We prepare it by suspending 45.3 grams of the purchased solid medium in a liter of water and shook it, and followed by the addition of 10 ml of glycerol. The resulting solution was autoclaved and cooled at approximately 50 degrees Celsius before poured into a sterile petri dish. For the neutron broth, 8 grams of the medium was dissolved in a liter of distilled water, shook and sterilized before autoclaving at 121 degrees Celsius. While the neutron agar, we dissolved 28 grams of the medium in a liter of the solution. It followed the same process by autoclaving at 121 degrees Celsius. So to isolate the Pseudomonas species from the soil sample, the sample that is soil was inoculated on the already prepared cetrimide agar, that agar we prepared the first. So using the strip plate techniques. So we inculate this plate at 37 degrees Celsius for 24 hours. Then this followed by the colony grown examination. In the plate, we observed that Pseudomonas colonies, which were appearing green in color, as we can see from the petri dish below, and then that we isolated the colonies and stored it in neutron broth, supplemented with 15% glycerol at -80 degrees Celsius for further analysis. So to check the antimicrobial sensibility test of this isolate we have isolated from cetrimide agar, so we took the isolate from the neutron broth and tested it against this antibiotic, that is tetracycline, ampicillin, chloramphenicol. And we also make a combination of ampicillin and chloramphenicol, making 4 plates. So we then pour we then pour the agar on 4 different petri dish. Then we inoculate the plate at 37 degrees Celsius for 24 hours. So we then examine the changes on the petri dish. We observe that 2 creamy colonies in a plate containing combination of the 2 antibiotics, that's the plate containing ampicillin and the chloramphenicol. So these 2 creamy colonies were then picked and stored in a neutron broth and incubated for 24 hours The neutron ball was shaken in a water for 2 days before taken to a research center in Zaria for plasmid extraction. The result for the antimicrobial sensibility is presented in this slide. When isolates were inoculated in a neutron agar with ampicillin, the antibiotic ampicillin, a scanty growth, which is circular in shape, was observed. Isolate also in neutron agar with chloramphenicol, a circular shape with scanty growth isolate was also observed. So, however, in a plate containing tetracycline, we didn't observe any growth, while in the plate containing ampicillin and chloramphenicol, we observed 2 colony growths. So these 2, as I have said earlier, these 2 colonies were then picked again for further research, that is plasmid extraction. So this showed that the isolate we isolated, that is the Pseudomonas species we isolated from the soil sample, is resistant to ampicillin, chloramphenicol, but is not resistant to tetracycline. The plate of this isolate are shown in this slide. Slide 1 is isolate in neutron agar with ampicillin. Slide B, neutron agar with the chloramphenicol isolate. Slide C, isolate in neutron agar with tetracycline. Slide D, isolate in neutron agar with ampicillin and chloramphenicol. The 2 colony we isolated from that resistant, we further extend them to plasmid extraction. For the plasmid extraction, the plasmids were extracted using about 600 microliter of bacterial growth culture was grown in a neutron broth containing an appropriate antibiotic and was transferred to 1.5 ml microfilters and spawned at 11,000 grams. The supernatant was carefully removed with a fine pipette and discarded. So to the resultant precipitate, 100 microliter of 7 times lysis buffer was added. This lysis buffer has compound of alkaline and sodium dioxide sulfate solution, and 1% of sodium hydroxide and 2% of sodium dioxide sulfate. So it was then inverted 5 times to get a clear suspension. Within a 2 minute, 350 microliter of cold neutralization buffer, that's 3 molar of guanidine hydrochloric, was added to the suspension, and content of the 2 were gently invited 3 times to ensure complete neutralization. Then the 2 were centrifuged, and the content of it were transferred to another column and placed into a collection tube and centrifuged at the same speed. This is a continuation of the plasmid section. So then, after discarding the flow-through, we added 200 microliter of endo-wash buffer to the dark zymospin in column and centrifuge again. This flow with the addition of another 400 microliter of the wash buffer and centrifuge before the content of the column was transferred in another 1.5 ml of microcentrifuge. So to the resulting mixture, 30 microliter of elution buffer was directly added to the column matrix and left stranded for 1 minute before it was centrifuged so that we can elute the plasmid. For the related centrifugal force of 11,000 was maintained, and the room temperature also was maintained. The procedure was repeated for the second colony. As you know, that we have isolated 2 colonies. So this is like an overview of the process. At first, we put in the pellet of bacterial cell, then we centrifuge, then we added lysis buffer, followed by neutralization buffer, centrifugation to pellet protein precipitate. Then we transfer the supernatant to that column. Then we wash it and elute the plasmid. So the plasmid we eluted with it were then extended to size determination through gel electrophoresis. As you know, that this electrophoresis is the method used to determine the DNA or RNA according to the size. So
Yunusa,
in this process,
Yunusa, can you hear me? You have 5 minutes
yes,
left. Okay?
Okay.
Use it wisely.
So in this process, okay, in this process, we prepare 2.0% agarose gel by dissolving 1 gram of the powder to 50 ml of tri-borate EDTA buffer in a 200 ml beaker. The beaker was heated and uncovered. Then the process followed with the addition of 3 microliter of ethidium bromide staining solution, so that this ethidium bromide solution can enhance visualization. The gel was then poured into a gel electrophoresis containing an appropriate comb to create a wall and allow to set at room temperature, cold before the comb was carefully removed. This followed with the addition of rhodium vapor to the electrophoresis tray up to the surface of the gel. Then the loading gel was added to our eluted sample. Loading gel helps to track how far the DNA sample has traveled. As you know that the DNA sample will travel toward the cathode ray because they have negative charges. So a 10 microliter DNA sample was slowly and carefully measured and loaded into the gel, and electrophoresis its DNA size marker, that is DNA ladder, was loaded along with our experimental sample. So the system was turned on and run at 120 volts for an hour. When the process was completed, the gel was removed and placed. The gel was moved and placed on the paper towel to absorb any extra rhodium vapor we have poured before. So the gel was then visualized in ultraviolet transilluminator. This is like an overview of the process. We make the hydrolysis, boil it, then pour it on the gel tray, remove the comb, then we forward the sample into the wells. Then we turn on the system, as I said, at 120 volt. Then finally we visualize this in the ladder, the system using ultraviolet transilluminators, so that we can see the site of our extracted DNA. So for the plasmid profile of the DNA, as I've said, was presented in the figure below. So after complete separation by electrophoresis, so the DNA fragments are visible according to the size. As you know that the lower size DNA will travel more faster than the higher size DNA. So from the graph, you can see that letter A shows the DNA fragment for the first isolate, that is isolate for our first isolate. But corresponding it with the DNA ladder, we observe that the approximate size of the DNA approximate size is 7 kilobyte. This approximate of 7,000 base pair. As you know, 1 kilo base pair is called 1,000 base pair. So letter B, which is isolate for the second sample, we didn't observe any plasmid from it. And letter C is positive control. This is the figure. This is our letter A, the plasmid of our first isolate. We didn't observe plasmid in our second isolate. This is the control. And the last one is the DNA ladder. So this conclusion, our conclusion will conclude that the plasmid of molecular weight of 7,000 base pair may be responsible for the resistance of the strain to the two antibiotic, that is, resistance of the strain to the ampicillin and chloramphenicol. Thank you for listening.
Thank you, Yunusa, for your nice presentation, and congratulations. And our next presenter is Kumar Paladyum. Kumar Paladyum is here, or Yasemin Kurt? Is not here, okay. Mamajan, Ovezova, Sulaiman Gülcemal is here.
Evet, ben buradayım.
Okay. Before the presentation, you have 15 minutes for the presentation. Okay. Use it effectively, and let me read the title. Disiptütüye imidazol 2 iliden ligandi içeren paladyum paladyum komplekslerinin sentezi ve katalitik uygulamaları.
Evet.
Evet, başlığınız bu.
Hocam,
İsterseniz
bir saniye Umar, if you could please stop screen stop sharing your screen. So Mamajan can share her.
Yunusa Umar, please stop sharing your screen. Okay, thank you. And Mamajan,
Evet.
Mamajan, in the beginning please, öncelikle bir kendimizi tanıtalım. Sonrasında sunuma geçelim istersen. Başlığımız
Tamam.
Türkçeymiş. Buyur.
Olur. Ben Mamajan Ovezova. Ege Üniversitesi Fen Bilimleri Enstitüsü Yüksek Lisans Fen Bilimleri Enstitüsü Kimya Bölümünde yüksek lisansını yeni tamamladım. İşte Sulaiman danışman hocam doçent doktor Sulaiman Gülcemal ile çalıştığım çalışmaları size kongrede sunacağım.
Evet, teşekkür ederiz. Buyur.
Biz teşekkür ederim. Ben teşekkür ederim. Share screen'den yapıyorum değil mi ama
evet.
Şimdi denebilirsin.
Tamam. Oldu herhalde.
Evet, tam ekran yapalım sunumu da.
Tamam. Evet.
Sağ alt köşeden, evet.
Başlayabilir miyim?
Evet, buyur.
Teşekkür ederim.
Rica ederiz.
Merhaba arkadaşlar. Bugün size bu kongrede 4-5D substite indizoli 2 iliden ligantı içeren paladyum komplekslerin sentezi ve katalitik uygulamaları konu başlığı altında hazırlamış olduğum sunumu size sunacağım. İlk önce katalizör nedir? Bunun tanımıyla başlamak istiyorum. Katalizör, tepkimenin aktivasyon enerjisini değiştirerek farklı mekanizma üzerinden yürümesini sağlayan maddelerdir. Katalizör her tepkime için özeldir. Yani bir tepkime için uygun katalizörü ancak deneysel yollardan bulabiliriz. Bir katalizörde, ideal katalizörde yüksek katalitik aktivite, yüksek katalitik seçicilik özelliğinin yanı sıra çevre dostu olması, atom ekonomisi ve düşük maliyetli olması, katalitik kararlılık gibi özelliklerinin de olması beklenir. Homojen katalizörlerde N-heterosiklik karbenler yaygın ligand olarak kullanılmaktadır. N-heterosiklik karbenler tanımını şöyle yapabiliriz. Heterosiklik yapısında en az bir tane amino substansı içeren karben türleri olarak tanımlayabiliriz. N-heterosiklik karbenlerin şu azot atomu üzerinde veya doğrudan imidazol iskeletinin bağlı olan, sırt bölgesine bağlı olan grupların uygun kombinasyonu ile kararlı olan N-heterosiklik karbenler sentezlenebilmektedir. Yaygın olarak kullanılan N-heterosiklik karbenler, bundan sonra NHC diye adlandıracağım. İmidazolin ikiliden, triazol ikiliden, imidazol ikiliden, benzimidazol ikiliden bu NHC'ler homojen katalizöründe yaygın olarak ligand olarak kullanılır. NHC'lerin sentezine ilişkin literatürde birçok yöntemler vardır. Ama en yaygın olarak kullanılan yöntemler azolyum tuzlarının in situ deprotonasyonu, imidazolidinlerin alfa eliminasyonu ve imidazolin 2 tiyonların disülfurizasyonu sonucunda serbest karbenler elde edilebilmektedir. Siz de ileride göreceksiniz ki çalışmalarımızda azolyum tuzların in situ deprotonasyonu yöntemiyle karbenlerin elde ettik. Azolyum tuzları da iki farklı yöntemle elde edilebilir. Bunlar inibizoldeki azot atomlarının alkilasyonu tepkimesiyle, diğer bir yöntemi ise glikoksal ve birincil amitlerin çok bileşenleri reaksiyonları sonucunda azolyum tuzları elde edilebilmektedir. NHC metal kompleksleri, NHC'ler kuvvetli sigma verici ve zayıf pi alıcı özelliğinden dolayı metallerle kararlı bir bağ oluşturmaktadır. Bu bağdan dolayı bu kompleksler reaksiyon ortamında yüksek sıcaklık, oksidasyon ve hidrolize karşı kararlıdırlar. Bağlı olan grupların, şu yani az önce de söylediğim gibi azot üzerindeki R üstü grupların ya da heterosiklik halkaya doğrudan bağlı olan Y üstü grupların doğru kombinasyonu ile yüksek derecede etkin ve kararlı NHC metal kompleksleri sentezlenebilmektedir. NHC metal komplekslerinin sentezi ve katalizdeki uygulamaları. NHC metal komplekslerin birçok yöntemleri vardır ama en yaygın olarak azolyum tuzların tepkime ortamında, yani in situ olarak protonsuzlaştırma, transmetalasyon, üçüncü yöntemimiz ise serbest karbonların doğrudan metal öncülleriyle tepkimesi sonucunda azolyum tuzlarından NHC metal kompleksleri sentezlenebilmektedir. Bu kompleksler çapraz eşleşme, transfer hidrojenasyon, polimerizasyon, hidrojenasyon, hidroformülasyon gibi birçok tepkimelerde yaygın olarak, katalizör olarak yaygın bir şekilde kullanılmakta. Heterosiklik paladyum kompleksleri ve karbon heteroatom bağ oluşum reaksiyonları, az önce de dediğimiz gibi bir eşleşme reaksiyonlarında NHCpG kompleksleri yaygın bir şekilde kullanılmakta. Suzuki Miyaura, Mizuroki, Hicks, Noguchi ve Agira gibi birçok karbon bağ oluşum tepkimelerinde NHC paladyum kompleksleri aktif katalizör olarak yaygın şekilde kullanılmaktadır. Burada substrat olarak alkil halojenürler kullanılır. Alkil halojenürler tepkime sonucunda çevre açısından atık oluşabilmektedir. Ketonların primer alkollerle alfa alkilasyon tepkimesi de bir karbon bağ oluşum reaksiyonlarına örnektir. Bu tepkimede substrat olarak alkil halojenürlerin yerine alkoller kullanılmaktadır. Alkoller daha ucuz ve kolay bulunabilir ve bu tepkime sonucunda çevreye zararlı olan atık değil de tepkime sonucunda sadece su oluştuğundan karbon bağ oluşum tepkimesi açısından ketonların primer alkollerle alfa alkilasyon tepkimesi önemlidir. Pepsi tipi paladyum NHC kompleksleri, Pepsi tipi paladyum NHC kompleksleri aktif paladyum sıfıra indirgenebilen paladyum 2 ön katalizörleridir. Pepsitlerin İngilizce bir terimi olup piridin ile zenginleştirilmiş ön katalizör hazırlama, kararlaştırma ve başlatma anlamını veren sözcüklerin kısaltılmasından oluşmaktadır. Bu kompleksin sentezi şekilde gösterildiği gibi paladyum klorür ve paladyum klorür baz ve piridin varlığında 80 derecede geri soğutularak Pepsi tipi paladyum kompleksler elde edilebilmektedir. Bu sentezi ilk olarak 2006 yılında Organ ve arkadaşları tarafından sentezlenmiştir. Ve günümüze kadar bu sentez yöntemiyle bu tip kompleksler sentezlenmektedir. Paladyum Pepsi komplekslerin yapısal özellikleri şekilde özetlenmiştir. Bu tip komplekslerin şöyle avantajları var. Kolay hazırlanabilmektedirler. Hava neme karşı oldukça kararlıdırlar. Çeşitli birçok reaksiyonlarda yüksek performans göstermişlerdir. Hidrojen ototransfer yöntemi. Hidrojen ototransfer yöntemi hidrojen ödünç alma yöntemi olarak da bilinir. Bu yöntem daha karmaşık moleküllerin sentezinde bir veya daha fazla ara basamakları birleştirerek tek adımda gerçekleştirmesini sağlayan bir önemli yöntemdir. Bu yöntemde substrat olarak gene alkoller kullanılır. Alkoller kullanılmaktadır. Katalizör tarafından substrattan hidrojenler koparılır. Dehidrojenlenmiş substratlar ikinci basamakta, ara basamakta aldol tepkimesi gerçekleştirilir. Daha sonra katalizör tarafından alınan hidrojenler üçüncü basamakta geri verilerek hidrojenasyon gerçekleşir. Hidrojen ototransferi yönteminin şöyle avantajları vardır. Moleküler hidrojenin doğrudan kullanımını gerektirmez. Substrat kaynağı olarak kolay bulunabilir. Alkoller kullanılır. Tek adımda gerçekleştiği için atom ekonomisi sağlanır. Ve substrat olarak alkollerin kullanıldığı için tepkime sonucunda atık olarak sadece su açığa çıkar. Bu da çevre açısından önemlidir. Deneysel çalışmalarımıza geçersek, deneysel çalışmalarımızda ketonların primer alkollerle alfa alkilasyon tepkimesinde katalizör olarak kullanılmak amacıyla 6 tane katalizör sentezledik. Bu katalizörler önce azot atomu sesim geliyor mu?
Evet, duyuyoruz hocam.
Tamam. Çalışmalarımızda 6 tane farklı katalizör sentezledik. Bu katalizörler ilk önce azot atomları ve hetero halka üzerine doğrudan bağlı 4 ve 5 konumlarını substitute edilmiş imidazolyum ligandların tuzlarından azolyum tuzlarının in situ titratonasyonu yöntemiyle 5 farklı Paladyum Pepsi tipi NHC kompleksler sentezlendi. Bir de transmetalasyon yöntemiyle PD NHC kompleksi sentezlendi. Burada amaçladığımız şey 4 ve 5 konumlarındaki bağlı grupların katalizöre nasıl bir etkisini görmek için 4 ve 5 konumlarına fenil, 4-metoksifenil, 4-klorofenil gibi farklı gruplar takıldı. Daha sonra sırf modifikasyonu yapıldıktan sonra azot atomu üzerindeki metoksi, elektron sağlayıcı metoksi grubu yerine elektron çekici trifluorometil grubu bağlanarak azot atomu üzerine modifikasyonu yapıldı. Daha sonra elde ettiğimiz katalizörü belirlenen seçtiğimiz ketonların alfa alkilasyon reaksiyonunda denemek amacıyla 6 katalizörümüzün aktifliğini inceledik. Burada gördüğümüz gibi bir de NHC ligandlarının aktivitesine komplekse nasıl bir etki ettiğini görmek amacıyla bis-karben kompleksini sentezledik. Bir de piridin ligandının bu katalizöre nasıl bir etkisinin olduğunu görmek amacıyla bispiridin paladyum kompleksini sentezledik. Bir de paladyumun kendisinin nasıl bir tepkime üzerindeki etkisini görmek amacıyla paladyum klorüründe kendisini katalizör olarak belirlenen tepkime üzerinde aktifliğini inceledik. Ayrıca katalizörsüz kör dönemi bazı varlığında tepkime gerçekleştirildi. Tepkime sonuçlarındaki veriler tabloda gösterilmiştir. Görüldüğü gibi en aktif katalizörümüz olarak sırt bölgesi yani 4 ve 5 konumu 4 metoksi benzil grubu azot atomu üzerinde yine 4-5 azot atomu üzerinde yine 4 metoksi fenil grubunun bağlı olduğu paladyum bağlı olduğu hepsi tipi paladyum NHC kompleksimiz aktif çıktı. Daha sonra optimizasyon çalışmalarını belirlemek için bu tepkimelerde bağ taraması ve çözücü taraması yani optimizasyon şartları belirlemek için farklı bazlar ve farklı çözücüleri kullanılarak tepkimeler yapıldı ama optimizasyon şartımız olarak fenilin 2 ml fenil metanol baz olarak potasyum hidroksit belirlendi. Aktif katalizörümüz olan C3 numaralı katalizörümüz %0,1 mol varlığında 6 saat içerisinde açık havada belirtilen reaksiyonun sonucunda ürün ketonlarımız elde edildi. Bu denemede farklı sübsteli ketonlar ve alkoller denenilerek 12 farklı türlü keton ürünü %63 ile %93 verimi aralığında izole verimler elde edildi. Bu çalışmanın...
Mamajan son birkaç dakikan kaldı. Toparlayalım.
Tamam. Bitti zaten. Bu çalışmada 5 türlü ligand olarak azolyum tuzları, imidizolyum tuzları sentezlenildi. Elde edilen tuzlardan ilgili 5 tane peptit tipi paladyum NHC kompleksleri sentezlendi. Bir de ayrıca biz NHC komplekslerin sentezi gerçekleştirildi. Elde edilen kompleksler ketonların primer alkollerle tepkimesinde katalizör olarak denenildi. Aktifleri etkisi araştırıldı. Ve 12 farklı dallanmış keton ürünü değişen izole verimlerle %63-96 verimlerle elde edildi. Bu çalışmanın peptit tipi NHC tipi katalizörlerin belirtilen reaksiyon şartlarında ilk kez denenmesi literatür açısından önemlidir. Çalışmam daha devam etmekte. Dinlediğiniz için teşekkür ederim. Benim anlatacaklarım bu kadar. Sorularınız varsa ya da tavsiyeleriniz varsa alabilirim.
Evet. Mamajan'a güzel sunumu için çok teşekkür ediyoruz. Çalışmaların başarılarında devamını diliyoruz. Evet, soru varsa alabiliriz.
Evet var.
Dinliyoruz, buyur.
Evet, buyurun.
Merhaba.
Merhaba.
Nasılsınız?
İyi, teşekkür ederiz.
Güzel sunumu hazırladığın için teşekkür ederim. Yani önceki slide hidrojen ototransfer.
Hidrojen ototransferi yöntemi.
Önceki slide.
Hidrojen şu mu?
İstersen ekranını paylaş, o
tamam.
şekilde arkadaşımız da görsün.
Tamam. Hidrojen ototransferi yöntemi.
Önceki slide.
Bir önceki.
Ondan bir önceki slide.
Bir önceki, evet.
Aha.
Yani avantajlar söylemişsin. Hava ve nem.
Evet.
Kaşık karalıydı.
Evet.
Yani hava ilgili değişiyorsa nasıl olacak?
Ya bunlar genelde karbonlar kararsız karbonlardır. Yani o yüzden paladyumu piridin ligandı ve ayrılabilir piridin ligandı ve halojenürler doğrudan bağlı olduğu için paladyum katalizörleri +2 katalizörlerdir. 2 değerlikli katalizör, kararlı bir katalizör. Ancak reaksiyon ortamına girince, tepkimeye girince paladyum 0,2 paladyum 0'a indirgenebilen bir liganttır. Yani havada nemde bozulmayan bir ligand, katalizör.
Teşekkürler.
Ben
teşekkür ederiz. Başka sorusu olan var mı? Evet, herhalde başka sorumuz yok. Tekrar teşekkür ediyoruz Mamajan'a. Şimdi
ben teşekkür ederim dinlediğiniz için.
Teşekkürler. Ekranını paylaşmayı durdurabilirsin.
Tamam.
Teşekkürler. Kumars Paladyum burada mı? Yok herhalde. Abdikadir Abdullahi is here. Abdikadir Abdullahi is here.
I'm here.
Okay.
I'm here. Can you please send me?
Okay. Our next title is the relation between the severity of illness and microRNA 4520A levels related to RAS, homolog enriched in brain target of rapamycin pathway in familial Mediterranean fever patients. Abdikadir Abdullahi, Volkan Sözer, Hafize Uzun. Abdikadir, you have 15 minutes. Please use it effectively. Okay, you can start now.
Okay, Bismillahirrahmanirrahim. I'm Abdul Qadir Hassan Abdullahi from Yıldız Teknik University. I will present my topic related to the microRNA by looking the relationship between the microRNA 4520A and the red motor pathway. So I will share my screen.
Okay, thank you.
Okay. Is it? Can you see it?
Yes, we can see.
All right. All right. So familial Mediterranean fever is a monogenic recurrent disease, which is an inflammatory illness characterized by recurrence of fever in a lungi with polyceratitis. It is very common among the population in the Mediterranean region, including Arabs, Armenians, Turks, and Jews. Nevertheless, it's also progressively found in several parts of the world, including Italy, Greece, France, and some parts of Southeast Asia like Japan. Some parts of the world like Africa, specifically Sub-Saharan African, there's no data available for that region, but mostly in USA and that regions that I mentioned before, there is new cases. The disease is considered as a rare disease, and it's symptomatic. There's always fever in the beginning, and there's abdominal pain, and in the severe case, there's what's called amyloidosis. And amyloidosis is a situation that the protein called amyloid A accumulates in the extracellular, so it causes in the last in the end stage of renal failure or liver failure. This genetic, it's inherited and it's genetically based on the familial Mediterranean fever gene, which is a gene that encodes the protein called birin. So the highest prevalence is those people who are living in the Mediterranean region. In the beginning of the symptoms, patients might feel swelling in the ankles and also abdominal pain, and it brings fever. And then at the end stage, it might cause renal failure. So in Turkey, we are dealing with the Turkish population, specifically pediatric patients in Istanbul Cerrahpaşa Hospital, rheumatology department with the Özgür Hoca. And so when we look at the children, there's a deformity in the skeletal system because of the infection of this disease can cause a problem in the skeleton of the child, or it may impair their movement and mobility. So chest pain, abdominal pain, tender and swelling of joints, pain and swelling of joints, muscle pain, and these are the main problems that we see. So when we see on the basic of genetics and on the FMF, the mutation of MEF gene, which is called a Mediterranean fever gene, is a pyrin protein which includes which is responsible for the induction of the activation of inflammasome, results in enormous production of numerous interleukin-1 production. This interleukin-1 is the cause of inflammation, and these frequent mutations related to M69-4, M6801, and so on. So the MEF gene was acquired in human myelomonocytic cells, defined in the genus pyrin to identify the regulated of microRNAs. And these microRNAs is protected to target genes in autophagy through regulation of Reb motor signaling. This is a ribomycin, and mTOR is a mammalian target of ribomycin. So when we see in the family, if the father and mother are both carriers, if they are carriers and then they get married to each other, their offspring, their children will be like 1 in 4 might be unaffected, normal person. But 2 in 4 most of them might become like unaffected carriers, the same like mother and father. But 1 in 4 always become affected. So this is the chance probability of being a deformed child or being affected child with FMF if your parents are carriers. So recent studies being conducted on mTOR signaling pathway suggested this pathway might be a potential participant in regulation of exogenous of pyrin protein and FMF pathogenesis. mTOR mammalian target ribomycin pathway, which remarkably triggered by Reb enriched in brain. Negatively regulating autophagy can potentially detect the hazardous and metabolic signals through beta regulation of proteins. Besides, it plays an active part on the inflammation and cell growth. So these are the regulators. If the pyrin is absent from your gene, then the inflammation of interleukin-1 is not controlled. In that case, if it's not regulated, then there's enormous production of inflammasomes. That inflammasomes will cause an inflammation. That's why we call it causes some immune response generated damage in the brain, in the lungs, in the kidney, and in the skeletal system. Lucidius also has analyzed his expression of the microRNA 4520A and associated red motor pathway. Studying concluded that comparative level of expression employed by makers were drastically deregulated, so due to pyrin mutation. So we see like when there's the level of expression of microRNA are always decreased because of the absence of pyrin. So they also suggested that Reb has a potential role in activation. That Reb we mentioned it has a potential to activate in its side the pyrin, and the pyrin is tyrine threonine kinase activation. So in the literature, in the bioinformatics, as we have seen, microRNA 40520, the capability of target of the gene which are involved in autophagy regulation, like red motor pathway, recent studies have been conducted based on the relation of amyloidosis with the regulation of autophagy inhibitors. So Reb initiates mechanism which hinders the initiation of complementary autophagy destruction of the reduced NLRB, which is not like receptor protein in the pyrin, and that the responsible mechanism of not like reversing might be like pathogen associated, might be nitrogenous, oxidative nitrogenous, or might be like destruction of the cell wall. So our assumption was to check and to examine the level of mTOR and the level of microRNA expression. But because of the coronavirus and because of the pandemic, we couldn't complete our research yet. We are in the data collection steps. So maybe in the next Congress, we might suggest, like, we might show you the results. But it's still our research are in progress as we are collecting the blood samples from the patients. But the assumption that we have taken is, like, as we see in the literature, mTOR increases and the level of microRNA decreases. So in that case, if we interfere with mTOR and this microRNA 4520A, we might able to understand the association between the pathogenesis and the severity of the disease. That might produce, that might be a good step point for getting some therapeutic goals in the future, inshallah. And that's the conclusion of my presentation. Thank you for listening.
Thank you, Abdulkadir, for the nice presentation. Is there any question?
Yes, good morning.
Good morning.
Can you hear me?
Yes, we can hear you.
Okay. First of all, I would like to thank you for the nice presentation. And I'm wondering if you are planning to do certain studies with the different departments, such as multiple studies, maybe not only effect of the drug, but also the design and the administration of the drug. Are you planning such studies?
The question belongs to whom? Is it for Abdulkadir?
Yes.
All right.
It is for you.
Okay. So what we are planning is, for now, to understand the pathophysiology of the disease. So part on, we are working on molecular system in that, specifically microRNA and its involvement in that activation of red motor pathway. And then if we understand the pathophysiology, there should be a way that we can say, like, if this happens, we can block this enzyme, or we can block this pathway by doing this. So it will be possible treatment for the future. But for now, only we are working to define the molecular and to understand the pathophysiology of the disease. Now there is a treatment which is called colchicine. Colchicine might prevent the disease from occurring. The symptoms might occur, but like in a mild way, because the patient might suffer the pain the time of occurrence, and it happens like every 2 weeks or every 1 month. So by taking that colchicine, it makes the patient to feel like less pain, but not complete cure. So what we are planning is, like, as long as the disease has no cure, we are planning to find a way that we can get a complete treatment. So in the future, we are planning, if we get to understand the pathophysiology, then we are going to go to a way that we can treat the disease. Thank you.
Thank you very much. Clear enough. And at the same time, I think it's a quite promising. Once you can define, everything can be dissolved by designing such a drug delivery system to specify the certain enzyme, or to block the certain enzyme that taking the role in that action. That's great. Thank you for explanation.
Thank you for your comment and your question as well.
Okay, Abdulkadir, please stop sharing your screen. Okay, thank you. And our next title is Nihal Tabasi is here?
Yeah, I'm here.
Okay. Our next title is Uptake of Circumin by Supported Metal Oxides, Mesoporous Silica Materials.
Yeah.
Nihal, you have 15 minutes. Please use it effectively. Okay, yeah, now we can start.
Okay.
Öncelikle kısa olarak da bir kendini tanıtabilirsin.
sharing the screen. Hello to everyone. I'm Nihal Tapası. Doktora öğrenci, pardon, İngilizce Türkçe fark etmez değil mi?
Nasıl istiyorsan.
Tamamdır. Benim adım Nihal Tapası. I'm doktora öğrenciyim. Kimya bölümünde. Filistin'den yüksek lisans bitirdim. Kimya anorganik bölümünde. Şu an doktora Ege Üniversitesi'de doktora bölümünde organik kimyada olarak çalışıyorum. O kadar.
Teşekkür ederiz. Tam ekrana geçelim.
Tamam. Geçtim zaten. Today we'll talk about a research that's related to my master thesis, uptake of curcumin by supported metal oxide mesoborous silica. I want to thank the committee of the 4th International Science Congress. I work with my professor, Dr. El-Nahhal, in El-Azhar University at Palestine. Today we'll talk about 3 main topics. I will start with the introduction. We worked about curcumin. Curcumin is the measured curcumins and the main bioactive component of Curcuma longa. Curcumin is a natural product. We take it from curcuma. It's a species. We use it in food. It gives a yellow color for our foods when we add it. Curcumin has received ample attention as a potential drug because of various pharmaceutical applications as antioxidants, anti-inflammatory, and anti-carcinogenic agent, anti-Alzheimer's disease, and anti-cystic fibrosis, anti-neoplastic and wound healing effects. The main 2 applications is anti-carcinogenic agent and anti-Alzheimer's disease. But curcumin have 2 formula, a keto formula and enol formula. The most effective formula is a keto formula. The major problem with curcumin, it is very poorly soluble in water, which reducing its effectiveness as a drug for the treatment of this chronic disease. We all know that if we have to take a drug, it must be soluble in water, so our body can take it in its metabolism. So that to avoid this problem, we use a mesoborous silica and its incorporation of metal oxide. The main advantages of these materials that mesoborous silica with high surface area and large pore volume and narrow pore size distribution are highly promised for drug delivery system, and these days it's used as drug delivery. By incorporation of metal oxide in mesoborous silica, it also has a promising field of research such as catalysis, optics, and drug delivery. By combination between mesoborous silica and its and metal oxide, nanometal oxide, we have a good drug delivery material. In our present research, we aim to use our fabricating inert mesoborous system with avoiding hazard materials, with also easy for loading and unloading curcumin when needed. We also aim to use our fabricating supported metal oxide. We use 4 different metal oxide and we use 4 different mesoborous silica. The 4 different mesoborous silica using different surfactants to prepare it. And the 4 different metal oxide was zinc oxide and copper oxides, calcium oxide and magnesium oxide. These metal oxides have no hazard effects and no toxic effect. on our parties, excepting copper oxide. Copper oxide have a small toxicity, but it's we can working with it. So we prepared this supported metal oxide mesoporous silica using impregnation methods. We aim to investigate the curcumin uptake percentage by different metal oxide supporting onto 4 different mesoporous silica using a batch method. The first step of our works was synthesis of mesoporous silica. As I said before, we prepared 4 different types of mesoporous silica. There have 2 different shapes. The first shape was hexagonal. In general, they take them Santa Barbara 15. We will use a surfactant from triplocopolymer family. It have a name of B123. We use this system to prepare our silica. We dissolve it using acid. We don't see this this area, but theoretical olarak, we can say how the surfactant turned into its specific structure. The second type of silica, we use 3 different surfactants, L81 and L61 and L31. These surfactants give us a lamellar silica, which is as a sheet. Then after synthesis of mesoporous silica, we synthesize the metal oxide encapsulated mesoporous silica using impregnation method. Impregnation method, one of the most the most perfect encapsulated method. In this method, we take a specific amount of metal oxide. Our percent, it was a 30% from the total material. At the first, we take metal oxide as a solution. Then our synthesized mesoporous silica was added under stirring until dryness. Then we drying our material overnight at 100 degrees. After this process, we have our encapsulated metal oxide. As I said, we use 4 different metal oxide: copper oxide, calcium oxide, magnesium oxide, and zinc oxide. After the synthesis of metal oxide encapsulated mesoporous silica, we make a complexation between a curcumin and metal oxide, and we study the uptaking of mesoporous silica for curcumin. It gives us a little amount. So we, I here want to view our curcumin metal oxide encapsulation. Curcumin metal oxide make complexation with metal oxide and give us this is formula. Here, this is a curcumin in solution. We use ethanol to dissolve curcumin. After adding the metal oxide as a solid material, it's precipitate and give us the ring of curcumin. Here with the other type of silica. To see this performance and to understand how this thing is working, we used a different type of characterization analysis using FTIR, Fourier transform infrared spectroscopy, X-ray diffraction, and thermal gravimetric analysis, ultraviolet. Here in the FTIR, we use it for curcumin. When we make FTIR after complexation, we cannot see curcumin because mesoporous silica have a broad peaks. When we make an XRD, X-ray diffraction, some kinds of this XRD related to some metal oxides. At A, we have encapsulated metal oxide in silica. B, it's related to curcumin. Pardon. To curcumin. To curcumin calcium oxide encapsulated. If we look here, we can see that some of these peaks was disappeared. That means the complexation was exist. Here is 2. Then we took it. Then we use thermal gravimetric analysis (TGA). Here it's related to mesoporous silica. It's also here for magnesium oxide. Here into calcium oxide. As we can see in thermal gravimetric analysis, every peak related to a specific area of decomposition of our materials. Here the loss of water, and then the loss of carbon dioxide, and in the last peak we have a condensation of silica. This is related to. After that, we make a curcumin calcium to for different type of silica. In the right, we have a curcumin complex with calcium with Santa Barbara 15, which is has a hexagonal structure. If we look here, we have a loss of water. Then a decomposition of curcumin was from 200 to 430 degree, and we have a 6% from loss of from the total amount of our materials. And this is for magnesium oxide complexation with curcumin. This is the curcumin, copper, and zinc. After that, we use UV visible. UV visible also takes for salzic curcumin, just for curcumin, which is related to A. Then with silica, if we notice here that silica does not make any absorption of curcumin, and its uptaking was so a little. Then if we make a comparison between a magnesium oxide, which is related to a blue, and the pink one related to curcumin calcium, to copper uptaking. Herein we can note that magnesium oxide have uptaking more than the copper oxide. In the right side, we make a comparison between calcium oxide and zinc oxide uptaking of curcumin. If we look here, we notice that zinc oxide have a little amount of uptaking of curcumin, but calcium oxide almost take a lot of curcumin, and a little amount of curcumin was exist. Herein we make a conversation between and a calculation for the uptaking of curcumin. If we notice about the same metal oxide for calcium, if we notice here the most uptaking was calcium oxide with Santa Barbara 15 with 96%. Then after that, we have a little difference between them. But when we make a comparison between a metal oxides with each other, we can note that calcium oxides have the most curcumin uptaking from the others, magnesium from the other metal oxides. After it, magnesium oxide is coming, and after that, copper oxide, and and at the little was zinc oxide, but zinc oxide have about 75% of uptaking. So finally, for curcumin metal oxide encapsulated mesoporous silica, UV visible analysis was the most perfect analysis, and curcumin uptake results show that all synthesized metal oxide encapsulated mesoporous silica materials were successfully formed curcumin metal complex with different uptake percent depends on the nature of metal oxide and mesoporous silica. It's found that calcium oxide with Santa Barbara 15 was the best for curcumin uptake with curcumin percent about 96. Thanks for listening.
Thank you, Nihal, for the nice presentation. And is there any question for the presentation?
Yes, I have a question.
Okay.
Thank you for a nice presentation. Actually, I have several different questions, but I don't know how many minutes left. If there is enough time, we can talk. First of all, have you decided to work with a different type of mesoporous silica nanoparticle apart from the Santa Barbara?
Yes, I want to work about something like that, and these days I work about a little project for this topic.
Okay. One more thing. Who is your PhD advisor?
I don't hear.
Have you already decided who is going to be your thesis advisor?
Nowadays, I work with my advisor was Derya Hoja, but we don't work about these topics because it have a lot of it hasn't a lot of information about this topic. We work about a catalysis, but this work I will work it as a special project.
Okay, we are preparing mesoporous silica nanoparticle as well, and we use them for drug delivery system and for several different purposes. That's why it interested to me. And you said that you made the uptake of the curcumin, but by which method? By using the chemical methods or physical absorption? How you attach the curcumin to the metal oxides?
It was a chemical
attachment
reaction because it's make a complexation between metal oxide and curcumin. But when we take it in our bodies, it will compost because of the pH. Curcumin at pH, acidic pH, make a relation, decomposition from metal oxide.
Okay. Have you studied the release of the curcumin?
I don't hear. I'm sorry.
Have you studied the release of the curcumin? Because you show the UV visible spectroscopy, and
yes.
Apparently the uptake it's good enough. Okay, well done, congratulations. But it's uptake or it's just made a complexation because of that the peak of the curcumin disappeared? If so, it's not going to work for your purpose. But if you studied the release of the curcumin and if you can calculate the amount of the curcumin, so that's great.
We studied the release of curcumin by adding HCl, a small amount of HCl. It gave us the reverse direction, which gave us methyl chloride and curcumin, a free curcumin.
Okay, but you have to keep in mind that if you would like to work for certain purposes, for human body, you have to calculate the pH carefully. Instead of the working under the acidic condition, maybe you have to keep in mind that you have to work on slightly basic condition or in certain buffers, such as the fact that PBS buffer can be one of the options?
We have no chance. We had no chance to make something in the body, but we make a little experiment about in vitro. In vitro, this is give us a good result, but it was in Palestine.
Okay, okay.
With my supervisor.
Okay, okay, okay. Thank you for your nice presentation. As I told you, we are working apparently in the one of the same project, because we have several different projects related to this topic. That's why it interested to me a lot. Thank you for your information. Thank you for the nice presentation.
Thank you, Hocam.
Bye bye.
Okay.
Bye-bye.
Okay, Nihal, thank you again for the nice presentation and for the nice explanations. Okay, please stop sharing your screen.
No, 1 minute, please. No, I don't know.
Is Dr. Kadim here?
Yes, I'm here.
Okay. Our next title is Determination of Inelastic Longitudinal Electron Scattering Form Factors in 58 Ni. Okay. And we can see you. Okay. You can start now. Please share your screen for us.
Hi, everybody. My name is Dua. I am from Iraq. I have a master's degree in nuclear physics. Today, I'm going to share with you a part of my master thesis regarding determination of inelastic longitudinal electron scattering form factor in nickel isotope. The aim of this study is to study the nuclear structure of nickel isotope by using electron scattering form factor. At the beginning, I will give you a brief introduction about that subject. Then I will go to the theory that used in this study. Then I will discuss with you the result and happened. At the beginning, now to start, we need to ask an important question, which is why electron scattering? Actually, there is two reasons that electron scattering are used to this study. First, the interaction is known since the electron charge particle and the proton in the nucleus is charged too. So the interaction is known as quantum electrodynamic. The second reason is the interaction is relatively weak, so it won't disturb the nucleus. There is two types of electron scattering. First, elastic electron scattering, which measures the radius, the thickness of the surface in the nucleus. The second, inelastic electron scattering, which I'm using here, is to study the transition properties for the nucleus. There is two types of inelastic electron scattering form factor, longitudinal, which I'm studying here, and the transfer, which is electric and magnetic. At the beginning, the main equation of the form factor is given by this equation. This is matrix element and this some correction. I won't go into detail with the equations. Just to know, this matrix element is divided into two parts, model space and corpolarization. Since the nucleus, as we know, has proton and nucleon, these protons and nucleon are distributed into a shell. This shell, a closed shell, called the corpolarization, and some few protons and neutrons are called model space. Each of these terms is consists of one body density matrix and matrix element. The one body is calculated from Oxbash, and these matrix elements are two methods are used in it, which is MSDI models, MSDI and M3Y interreaction. In this study, in this M3Y potential, we have not a good agreement with experimental data, since just 2 peak has appeared. So we headed to MSDI, which gave us the exact shape of the nucleus, especially when core polarization effects are included. Yet it needs more work to do, more methods and more effective charge to use. Thank you for listening.
Okay. Thank you for the nice presentation. And is there any question? Okay. Okay. Our next title is for the Umar's Paladyum is here. Kumars, are you here?
I think hocam he didn't attend.
Katılamadı mı? Yok mu?
Evet hocam katılamadı. Hiç görmedim.
Peki. O zaman bekleyelim mi biraz daha, yoksa yani gelir mi? Gelirse bekleyebiliriz. Sıkıntı yok, ama gelmeyecekse beklemeyelim.
Hocam.
Ama yok herhalde, hiç ses çıkmadı sabahtan beri.
Evet. Evet. Sabahtan gelmedi.
Okey, peki. 5 katılımcımız için, 5 başlık içinde güzel sunumları için teşekkür ediyoruz. Katkılarından dolayı, bilime olan katkılarından dolayı kendi teşekkür ediyoruz. O zaman bu session'da bitirebiliriz. Doğru mudur?
Tamam hocam.
Peki. Ben katılımcılara çok teşekkür ediyorum. Güzel sunumları için ve katkıları için. Sorusu olan veyahut da eklemek istediği bir nokta olan bir arkadaşımız varsa dinleyebiliriz.
Teşekkür ederim.
Peki, herhalde o da yok. O zaman hepinize iyi günler diliyorum. Ben teşekkür ediyorum.
Teşekkür ederim.
Teşekkür ederim.
İyi günler.